Hey, I just wanted to quickly plug that if you guys want any tutoring, some new slots have opened. If you look in the description, there's a link to my physics and maths tutor profile in which I do tutoring on there. Hello everybody, today we're going to be going through the OCR A-Level Biology A 2022 Paper 2. So many processes in the body are controlled by enzymes. Which of these options are an extracellular process controlled by enzymes? So extracellular means it's outside of a cell. If we look at each of these options, that does happen outside of a cell. Digestion of a pathogen inside is inside a cell. DNA replication is inside the cell. Movement of vesicles from the Golgi apparatus to cell membrane is inside a cell, so A. So now it says many industrial processes use immobilized enzymes, which is not an advantage of using them rather than free. So if you have an immobilized enzyme, imagine this is like a tube, and I've got the substrate moving in, and these are immobilized. It means that all I have to do is pass the stuff through the tube, and it will react, and these will stay in place. So if we look at our options, we need to figure out what the benefits could be. That is an advantage, because the enzymes are staying there, they're not being passed out in the mixture. Enzymes remain active over a wide range of temperatures. That's also true as well because they're stuck in place, so they don't have to, they can sort of resist a bit more temperature increase before it's um, starting to denature and stuff like that, or vice versa. So, for example, if the temperature is quite low, they don't actually have to whiz around and whack into and collide with the substrate to form an enzyme-substrate complex. They're in place there. All you have to do is just pass it over, so it will collide anyway. So it's not that one. Setup costs are low. Setup costs are low. Well, no, it's quite expensive to set that stuff off. It's way easier to just kind of use a free floating enzyme thing. So it's probably C. And the product is not contaminated by enzymes. Well, that is an advantage. Okay, so which of the following changes of lifestyle are or is likely to reduce the spread of disease that is transmitted by the droplets of moisture? So if we look at the options we've got, so clean drinking water is probably not really going to do that because this is talking about droplets of moisture. So it's talking about things like, for example, coughing and air spreading the, infective stuff. So it's probably not you. Yes, if you have fewer people living in the same house, because that means you're less in contact with other people and who are sick and maybe cough and breathe the stuff out at each other. And also, housing with improved ventilation as well means that all the air is cleared out. So that means it has. So ash trees are an important part of a British landscape. In 2012, a fungal disease known as ash dieback arrived from the UK from mainland Europe. Which of the following could explain how an ash dieback could have reached the UK from mainland Europe? So in fungal diseases, the options are that they can be spread through the wind, like the spores that are made, and they can also be transferred between infected plants. You put an infected plant near another one, or you could put an infected plant in the soil and then it dies and it leaves behind the spores. Now the thing is here in the question is it's telling you that this arrived from mainland Europe, so it's got to be ones that relate to this, if that makes sense. So for example, spores carried in the wind is possible because, for example, they could have brought an infected plant and the spores traveled in the wind. So that one's pretty all right. Then it says young diseased trees were imported and then planted. That's also possible. Contaminated soil from previous infected crop. Well, they've told you the question's told you in the context that it's arrived from the UK. Sorry, from mainland Europe to the UK. So there wouldn't have been any previous ones in the past. So it can't be that one. So that one is B. So number 5 says what chemicals produced by plants in response to attack by pathogens. So amylopectin is part of starch, and that's a storage molecule for glucose. It's not you. Amylose is also part of the starch, which is the storage molecule for glucose. So you're out as well. So that's branched, that's non-branched. So callose is a polysaccharide that's released by plants whenever they have sort of a need to recognize an attack of something from a pathogen. Scientists don't really know how it works, but what they think it does is it deposits it in the cell membranes and cell walls next to infected cells. What they do is it kind of barriers off that infected cell and stops it from spreading out into other places as well. So callose is one of them, and cellulose is beta glucose in a long polymers, and that causes them giving them sort of strength and stuff in cell walls and things like that. So it's not really a thing that's produced by plants and attack cells. It's not that either. So it says different types of microorganisms can act as pathogens. Which of the diseases caused by a pathogen without mitochondria in its cells? So if you look at all the options we have here, so first of all we've got black sigatoka in bananas. So black sigatoka is caused by a fungus, and fungi do have mitochondria. So now it says late blight in potatoes. So late blight in potatoes is again by a fungus like Protoctus, which again has mitochondria, so you're out. Now if we look at ring rot in potatoes is caused by a bacteria. Bacteria do not have mitochondria because they're prokaryotic. They don't have double membrane bound organelles, and that includes mitochondria, so C. And ringworm in cattle, if we wanted to go further and just say that is a fungus. On a biology field trip, a student calculated that an area of ancient woodland has a Simpson's index of diversity of 0.85. Which interpretation of a Simpson's index diversity is correct? So the way you calculate it, it's d and equals 1 minus sigma n squared over n, like that. Now, if you have a really high biodiversity, as in it's like, infinite, then it would be 1. And if you have absolutely no diversity, it's 0. So the scale is always 0 to 1. So this is quite close to 1, so its biodiversity is high. There you go. Now it says, which statement about the secondary structure of a protein is correct? So beta-pleated sheets contain beta-glucose. No, they don't. This happens due to oxygen, hydrogen, nitrogen atoms of the amino acids interacting with each other. So that's out. B, so disulfide bonds, I call them bridges, just that routine. Disulfide bonds are not involved. That is correct. They're not here yet. That tends to be the tertiary structure. And then each single polypeptide is either an alpha helix or a beta pleated sheet. That's not correct because it doesn't have to exclusively one or the other. And then also it says hydrogen bonds formed between the R groups of different amino acids. That's also incorrect because in the secondary structure of a protein, the variable groups are not involved at this stage. So that's a tertiary thing. So it's got to be B. So now it says cellulose is the main component of plant cell walls. What's not a property? So remember that cellulose, they form long, long, long fibers like that, and they're very, very good at just sort of being very rigid and hard. So you got to think of it. Imagine you've just got this rigid rod, and you've got to think what's going on. So high tensile strength means you can pull on this and it won't break apart. That is true. They do have that, so that's fine. Inflexible is also true because it's rigid. It can't bend, so that's all right. C, insoluble in water. Well, it's got glucoses in it, and it's got abilities to make hydrogen bonds, so and it can polarize water, and it's polar. So that's also that is a property that they have. And then 9D, resistant to digestion by enzymes, is incorrect. You can digest, we can't digest it as humans, but other animals like cows and stuff can, so D. So now it says glycogen is a large polysaccharide. Which option describes the structure and function of glycogen? So if we look, it says 1 to 6 glycosidic bonds are more accessible to the enzymes than 1 to 4 glycosidic bonds, so energy can be released more quickly. So with glycogen, it's branched, and that's what gives it its more accessibility to mean you can hydrolyze more glycosidic bonds and release energy, so that one's out. Bonds between beta-glucose residues, well, no, it's not made of beta-glucose, so that's out. If we look at C, exposed OH groups means that glycogen is soluble. So if you look, exposed OH groups means that glycogen is soluble. Well, admittedly, yes, while OH groups mean that something could be more soluble, glycogen is a storage molecule and we want it to be insoluble. One of its properties is that it is insoluble, and the reason is because there's not enough OH groups in it exposed to be able to do that. So it's actually incorrect because, again, we don't want it to be soluble. It's an energy store. So the last one is short branches allow for more energy release in a small space. It's got to be that one, and that makes sense because it's actually compact and really stuffed together so you can fit more energy in a smaller volume. So now it says which nitrogen cycle reaction is carried out by nitrobacter. So nitrobacter converts, and what if you want to say the proper word, oxidizes nitrites into nitrates. So if you remember that nitrates is NO3 minus, nitrites is NO2 minus, now you can look for the option that has those two on either side, which would be this. Nope, because that's the other way around. So there you go. Some students incubated plasmid DNA with a restriction enzyme. After 24 hours, they used gel electrophoresis to analyze the products of the incubation. Which option shows the correct procedure for gel electrophoresis? What you want to do is you want to stick the sample onto some agarose gel. You want to then put in a voltage and leave it for some time, and then you need to be able to actually see the bands. You need to put some sort of stain on it first. So otherwise, you're just not going to be able to see anything. And then what you want to do is you want to either visualize it. So, for example, if you use like a fluorescent marker or something, you want to see you want to put UV light on it. If it's radioactive, you use an X-ray or some way of visualizing it, and then take a picture. So for the overall steps is, again, put your stuff in the well, run a pull of a voltage or like an electric current through it. Then you wait for it. You give it some time, and then you put something on it to be able to actually see it, and then you take a picture or whatever it is you want to do after that. So whatever one of these follows those steps. So load the sample on the gel, apply voltage for a time, photograph the gel, but we wouldn't see anything, that's that. And then load the sample onto agarose gel, apply voltage, stain the gel, and then photograph it, that's B. You can see it once you've stained it. So now it says which molecule shows ATP. So remember, ATP is adenosine triphosphate. So whichever one of these has 3 phosphates is going to win. Unfortunately, all of them do, so we're a bit stuck. So now we need to look for adenosine triphosphate. So think about adenosine, what's in it? So an adenosine triphosphate, it's got 3 phosphates, which you just said we can't really use because all of these unfortunately do. But then it has an adenine, bonded to a ribose sugar. So we need to figure out which one of these is a ribose sugar. Now, ribose will have, so ribose, if you, at the bottom of a ribose molecule, you'll always see these OH. If it's deoxyribose, then it'll be an OH and then an H next. So whichever one of these has got 2 H's on the bottom will be our ribose. So that one's out, So we're stuck between C and D. So the next bit to look for is adenine, the adenosine part. The adenine base is that pyrimidines, for example, like thymine and cytosine, they're smaller bases. So they only have single carbon ring structures, whereas purines are the larger ones, so adenine and guanine, and they have double carbon ring structures. So this is your ribose sugar. Those are your 3 phosphates. This has got to be the base. That one has only got 1 ring structure there. This one has 2. So by the process of elimination, that's D, and that is adenine. This one would be, that one's thymine. So that one's out, it's D. Evolutionary relationships can be determined by comparing certain biological molecules between species, which is commonly used to determine evolutionary relationships. So the two most that we use in this circumstance is cytochrome C, which is a protein used in respiration, and ribosomal RNA. It's because what happened with all the sort of proteins over time and stuff, they slightly vary between organisms, and but some of them are like really conserved. They'd stay like pretty much almost unchanged entirely among species. So there's those ones you want to pick instead of the ones that vary too much, because the problem is if they vary too much, that suggests that these organisms are more different than they should be. The reason that we have these varying levels of like different amino acid sequences and stuff in all the other proteins is that just because the variability where it's actually changing is probably outside of the part where the enzyme, for example, its active site, or like a protein or receptor is actually active, the bit where things actually take place. So it's pretty normal for them to be quite different actually, and still yet work, if that makes sense. Like one organism's amylase might differ from mine, but still have the same active site. So the two that nearly always are really not very changed very much are going to be cytochrome C and ribosomal RNA. So collagen's messenger RNAs actually vary too much. The reason we don't use cytochrome C is because it's tiny. Like that's just a tiny sequence for one singular like gene, and one singular protein. So that one's out. It's D. D would give us a very nice long sample. You can see lots of differences. Whereas this is kind of teeny. It's like implying that, like, for example, I'm making this up, but imagine we had a 3-base sequence and they were identical. That's like me saying, okay, these things must be the same thing because there's 3 bases the same. It's better to have like a gigantic long one because then you have more certainty in what's going on. Because if I have like, I don't know, billions of them and they're all huge amounts different, then you're like, okay, these are pretty different organisms. So you want to choose something that's conserved, but also really, 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 really long as well. So competition is an important factor about determining population size. Which statement about competition is not correct? So competition between two species can result in the extinction of the less well-adapted species. One of that is correct. That's natural selection, so that one's out. Competition occurs between individuals of the same species. That is possible. I mean, humans are another example. If you have an identical clone of you that lives the same life as you, and you both go downstairs to grab a box of X special K and start scanning it, and you're fighting over it because you have a limited supply, that's competition. There we go. So then the next one is predators only compete with their own species. That's not true. Every organism in the universe either can compete with its own species, but others as well. So it's C. And then species with overlapping niches will compete with one another. That is true, they do. So, see. So hemoglobin is a globular protein, and then figure 16.1 shows the structure of a hemoglobin molecule. So there are 4 levels of protein structure. Name the bond in the primary structure. So the primary structure is the sequence of amino acids, which are made by condensation reactions to form peptide bonds. Name the structure labeled X. It is heme, which is kind of where all the exciting stuff happens next. So globular proteins are roughly spherical in shape. They can also have additional things like, for example, prosthetic groups and stuff, but the one that kind of makes them more specific is the fact that they're spherical. It's always good in biology to sort of go through all these little concepts and think, what is the most specific that screams this? What one-liner says it's definitely this, for example? So if I see spherical protein, I'm thinking globular because that's the most like sort of screaming it out to me kind of thing. So sickle cell disease is a genetic disease that results from a substitution mutation of one of the genes that code for hemoglobin. Figure 16.2 shows part of the mRNA sequence that codes for normal hemoglobin and the corresponding sequence of amino acids. Figure 16.3 is a representation of the genetic code, so G, C, A. Let's have a look what the missing ones are. So if you look at 16.3, we've got ACU, so ACU, that's threonine, threonine. And then if we look at the other one, what's that? CCU, so CCU, proline, so it's threonine and proline. I'd say read that. Cool. Outline the role of RNA polymerase in the production of the mRNA sequence in figure 16.2. So it's not asking about like every single other step, about like complementary base pairing and all of this stuff. It's asking you, how did it make that sequence directly? So what does it do? It joins RNA nucleotides via condensation reactions to form phosphodiester bonds between them. So it's between a nucleotide and the next nucleotide next to it, not between the two, like for example AT here and AT and CG. That is complementary base pairing due to hydrogen bonds. You're not joining them like that, you're joining them this way. So make sure you make that really clear. So in sickle cell disease, the hemoglobin contains the amino acid valine in one of its positions normally occupied by glutamic acid. State the base sequence of the anticodon of a tRNA molecule that brings valine to a ribosome. So there's a multi-step thing we've got to go on and do here. So if we look at the genetic code, Where's valine? Valine could be. Where is it? So hold on. Where's glutamic acid, first of all? Sorry, glutamic acid is here. And they've told you it's a substitution mutation in one of the genes. So one of those letters is changed and it becomes valine. So you've got to do quite a few steps together. So if you look at GA, all the GAs, we can get GA. Where are we? GA. Where's valine? GUG. So it's probably GUG that was what did it, right? Because GAG, we're getting to valine. Something's got to have swapped GUG. GUG is valine. However, this is on mRNA. tRNA is going to have another anticodon to this. So it's going to be complementary. So it's going to be the other opposite. So it's going to be CAC. So it's just important to read the question and use the whole context. So in sickle cell disease, the mutated hemoglobin has a reduced ability to carry oxygen. Some gene mutations do not affect protein function. Use figure 16.3 and levels of protein structure to explain why some gene mutations do not affect the function of a protein. Please don't say active site and all of this kind of stuff. If you're talking about hemoglobin, if you're going to say that, you're talking about an enzyme. So what is it asking you to do? The objective is usually I do this to help me figure out directly what the question wants is, protein unaffected is the end. Sorry, oops. Unaffected is the end thing the question's told us. Like, look at the facts of the question and then explain it. They've told you that the protein is unaffected, and you can have some mutations that do that. That seems really goofy, but this actually helps you sort of frame your mind. Some mutations mean a protein's unaffected. Then you're like, oh yeah, it's all about that, genetic code degenerate and all that kind of stuff. So now we have to explain why that might be the case. So the genetic code is degenerate, meaning multiple codons or base triplets code for the same amino acid. But it says to use that figure, so we're going to use that figure and just come up with one. So, for example, if you look where, here, GAA, well, if you had GAG, GAA and GAG make glutamic acid. That's your first point. Now we need to think what else. So some mutations don't affect the protein. It's not only just that. It can be that sometimes you can still have amino acid substitutions, but it might not actually change the tertiary structure. The hemoglobin function and a lot of other protein functions depend on its tertiary structure. So what could happen is so I'm going to write AA for short because I'm lazy, but you write the whole thing. Substitution may not affect tertiary structure, which hemoglobin depends on. As a result, it's still functional. Now, the other thing as well is that you could have an AA that's substituted, but it has similar properties. So that means the secondary and tertiary structure may be unchanged. So what I mean by that is that they still might fold and coil in the same way and form the same hydrogen bonds and all of that kind of stuff too as well. What else could it do? Well, if we looked at other proteins, it might just be that the mutation may change the amino acid sequence and bonding, and structures like tertiary and so forth. It could be that it's away from the site of action or function of a protein. So any protein in the world, generally, it could be that it's affected somewhere else. So if we look at this enzyme here, and this is its amino acids, right? That's the active site there. Maybe there's a substitution that changes its shape on the other side. Still works. So, doesn't matter. So it might be that as well. So some students investigated the effects of time on the growth of bacterial populations. They produced a large flask of bacterial culture. They divided this large culture into a number of smaller flasks containing 50 centimeters cubed of culture. They then incubated the smaller flask at 20 degrees for up to 48 hours. Every 4 hours, they removed one of the flasks and counted the bacteria, and then they recorded the total number of bacteria and the number of viable bacteria in each of the flasks. So when growing them in culture, it's important that aseptic technique is used. State why it's important for that to be aseptic. It's to prevent contamination by unwanted organ. Next, the students prepared the culture by adding a suspension of bacteria to a flask containing nutrient broth. So list two precautions that should be taken when preparing a bacterial culture in order to ensure that the procedure is aseptic. So it's not asking you about make sure this is, the pH is controlled, blah, blah, blah. They're asking you, what are you doing to make sure that you're not, adding contaminants? So for example, you would use sterile pipettes or other specific equipment because you want to absolutely make sure that they've been autoclaved, or for example, you can heat them really high, things like that. As well as that, you can use, for example, you can disinfect any surfaces that you're working on as well. Other things you could do is you could have a Bunsen flame nearby, and that makes the air move upwards, so any airborne pathogens will start to float up and away. As well as that, you could probably use something like you could put like a cover on top of whatever it is, like the bottle, but it needs to not be airtight, or you can start getting anaerobic bacteria. But the reason is you can, that's good because it can stop contamination getting in as well. So there you go. So when counting the number of bacteria, the students performed the serial dilutions of samples removed from each of the small flasks. In each one, they removed 0.1 centimeters cubed and added to 9.9 centimeters cubed of water. To estimate the total volume of bacteria, they used the light microscope to count the number of bacterial cells in a 0.01 centimeters cubed sample in the final serial dilution. To estimate the number of viable bacteria, the students spread this much of the final serial dilution on an agar plate and then counted the number of bacteria they'd grown after 24 hours. The students shook each flask before they started counting them, explain why they did that. So if they just had a flask chilling here and all bacteria is at the bottom and they took a sample from there, there's no bacteria in that. So it's just to evenly distribute everything. Evenly distributed. Generally speaking, if they're ever shaking or inverting things, it's to try and evenly distribute stuff. So if you do chemistry, it's the same for the volumetric flask bit where they turn it in and out. Invert it, it's to mix it, but also to evenly distribute things. So it can be more difficult to count bacterial cells using a light microscope than it is to count human cells. So just one reason why bacterial cells are hard to count with a light microscope. They're just smaller. That's pretty much it. Now it says in one 0.1 centimeter cube sample, the students counted 52 bacterial cells under the microscope. Describe the calculation steps the students would need to take to estimate the total number of bacterials in the small flask. So just from before, I'm going to just collect that they had that 50 centimeter cube culture, and from that culture they then did all of this stuff. So this is so they found 52 cells in 0.01 centimeters cubed. So they got 52 cells in 0.01 centimeters cubed. However, this whole thing has 10 centimeters cubed. So again, we're assuming that that was what we found in that small teeny bit, but in this in this 10 centimeter cubed sample, they're evenly distributed. So we need to scale it up to, so you would want to scale up to 10 centimeters cubed. So what you would do is you would do 52 times 10 times 10 times 10, because it's 10 cubed. And again, it's just describing the steps. You don't actually have to do it. It's just describing them. So then the next thing is this. Remember that this is a sample that they've taken 0.1 of that original 50 and added it to 9.9 of water. If you do 0.1 over 9.9 plus 0.1, that's 0.01, right? As a percentage, that's 1%. So that means they've diluted it by 100. So as in, like, this is 100 times less concentrated than the original thing. So therefore, you would want to, get like, as in, undo the sort of dilutions. So you would factor in serial dilutions. So every single time they're making it 100 times more dilute, which means this would drop by 100 each time. So what you would do is you just do times 100 to the power of n, where n is the number of dilutions. So you can work backwards like that, and then remember that that's in a that's in a 10 centimeter cube sample. So in order to figure out how much is in a 50, you would just times by 5 to get the 50 cents. It's just undoing their stat. So then it says the graph shows the students' results. I'm just going to have a quick look at what's going on here. So we've got viable and total. So total is the dotted line, and that's fine, and viable's over here. So that's whatever. And I can see here they're using a logarithmic scale as well, 2, 4, 6, 8, 10, and so forth. And that's time. Cool. So explain with reference to the graph why they use the logarithmic scale. If we didn't use a logarithmic scale, we'd be dealing with, for example, if we looked at here, like if we looked at 10, that'd be 10 to the 2, that's 100. And then if we looked here, that's 10 to the 12, which is like bloody gigantic. What's that? 10 to the 12 is a billion. Whoops, why did it take me so long to do? Yeah, so it's a huge range. So you'd have to have a gigantic piece of paper. So the range of values too big to represent on paper, and it's always good to explain using an example, e.g. 2 would be, log, I should say, 10 to, would be 10 to the 2, which is 100, and then, for example, 12 would be, 1 times 10 to the 12, which is gigantic. So now it says, calculate the percentage decrease in the viable population between 40 and 48 hours. So if we look at where 40 hours, cool, so that's what? That's 12. Sorry, it's asking it in the viable population, so we're going to look for where 40 is. Why is it not drawing a straight line? What's that? That's like a 9. So the log 10 number is 9 for the first one. And then 48 hours, it's not doing it again, is 6. So the other number is log 10, 6. So to undo a log, you just need to raise it to, so this was your power, and that's the base you're going to do. So that would actually be 10 to the 9. And we're looking for the percentage decrease, so we're going to work out the difference between the two. So that's going to be 10 to the 6 for the other one. You need the actual numbers, you can't work the log numbers. And then we're going to do it over the original, which was 10 to the 9, and then times by 100. If we do that, we should get 10 to the 9 minus 10 to the 6 over 10 to the 9 times 100. It should give you 99.9. Whoops. It says, explain with reference to the graph the decrease in the population of viable bacteria between 40 and 48 hours. So if we look at what's, so again, it's just saying the bacteria, the viable ones, decrease from 40 to 48 hours. We need to figure out the why, and then we need to explain the how. So if we look at this graph, we can see that from 40 to 48 hours, I mean, we also just worked out as well, it's dropped pretty heftily. So we need to explain why. So we need to think in our heads, why on earth are they dropping? There's nothing in there that's killing them or destroying them or anything like that. They're just simply chilling in a culture and now they're dying. What must be going on is that the viable bacteria are beefing with each other, they're competing with each other, right? So they must be competing for each other for the same things. So it must be things like nutrients, like, for example, glucose or oxygen, things like that. So there's actually less O2, so, less respiration, and then they can die like that. As well as that, you can see here that this total count stays pretty stable, and then that's going down as well, so they must be dying. So the total count after 40 hours is higher than the viable. So that must mean that they've got to be dead, because otherwise, where else, like, that should surely drop as well. So that means they're actually dying. That supports that. So then we need to think, well, why are they dying overall? Because if they were dying at a balanced rate, it kind of stay the same. And if there was less death, if there's less death and replication, it kind of crawl up like that, and that would also crawl up too. But they're staying the same. So what's happening is that more are dying than surviving. So more death versus replication, because as they go higher and higher in number, remember that they're replicating. And then another reason that could be the case is that there's more waste products as well. Another thing you could say is you could actually say in price specific competition as well. I probably should spare it. So it's just using the graph to explain that kind of stuff, and we can see there as well that if this is the long-term number of bacterial cells, if it's going up, they're growing. If it stays the same, they're not growing. If they're going down, it means they're more dying than growing. Or replicating, sorry, not growing. The Masai Mara is a grassland ecosystem in East Africa with a large range of wildlife. The human population in the area is increasing. Increasing human population could affect the biodiversity. The wildlife in the Masai Mara includes several large animals like gazelle, zebra, and wildebeest. Domestic animals such as cattle, sheep, and goat are farmed by humans to provide food. Graph shows the population of some large animals in the Masai Mara between these two timeframes. Fine, so we've got the estimated population, we've got it in big numbers there. And we start in 1975 going and so forth. Wildebeest is this, gazelle's that, blah, 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 blah. I think it's always good when you get these tables to just have a quick look and look at each thing one by one, what's happening trends-wise, and then compare with and after as well. There's a lot of lines here, but it just doesn't hurt to do. So wildebeest kind of goes down, and it kind of keeps going down, but it drops faster here than it does here, and then it kind of levels off. So its numbers go down fast and then slow down, whatever. If you look at gazelle, it's kind of hard to see on this. It's sort of not dropping too much, and then all of a sudden between 1980 and 95 it drops a bit more sharply, and then it stabilizes again. And then zebra, what's going on here? So zebra it drops kind of sharply and then it levels off. It's kind of stable. Goat and sheep, it's kind of all over the place, but then it starts to climb up as well in the 1995 onwards. And then cattle, it's sort of stable, drops sharply, and then kind of goes up as well. So then do comparisons. So if you notice, I'm just looking at each line one by one, and then I'm looking at them together, like combining, this versus the others. I can see already that these are starting to climb up beyond that point, and that's starting to go down. And possibly, this is just me thinking about possible things, is that these are going up and they're what, goat, sheep, and cattle. It's probably that the humans are farming them and they don't really care much about the other two. Possibly, I don't know. Then it says calculate the rate of change in the wildebeest population between 1975 and 2007. So if you look at 1975 and 2007, 7. So the numbers that we're going to need are the change in them. So if we're looking at wildebeest would be what? 40,000. Sorry, minus 40,000. Right? And if it's a rate of change, what that means is how this changes with this. All right, so it's in the change in this over the change in that. So it's time. So we need to figure out how many years it's been between 1975 minus 2007. If you do that, you should get 110. Have I misread? Oh, I've misread it. Sorry, it's 30,000. 140,000 minus 30,000 over 1975 minus 2007 gives you minus 3437.5 roughly, minus 37. But then we kind of want to stick to the number of significant figures they've given us. So minus 3,000, right? It's because those are all two significant figures. You don't want to do more than the question's asking. So the reason that we need to keep the minus in is because it's the rate of change, and if it's decreasing, that's negative. All right. If I said to you, my weight has changed over last year, you don't know if it's gone higher or lower. But then if I said, my weight went from, I don't know, 70 to 60, then it's a negative, it's decreased. So that's a negative rate of change. So that plus or minus tells you how it's going, if it's going up or if it's going down. So it's really important you keep that in mind. So then it says the graph has been used to support the claim that rising human population in the Masai Mara has a negative impact on the biodiversity in the area. Use evidence in the graph. So I've already looked at the graph and sort of made sense of it, and it's already starting to come to me. So again, when it says use evidence to evaluate it, you need to basically say things that support it and then what limitations there are from drawing that conclusion from here. So we need to first of all identify what their conclusion is. So again, I like to do like a start and finish. That always helps. I've told a bunch of people that I teach this and they find it helpful. So what they're saying is they're saying that I always start with the outcome. So you're going to get decreased biodiversity, and then you need to think what the direct thing is impacting it. Don't just say, oh, well, the graph shows this, blah, blah. That's not the point. Something is directly causing that, is what they're concluding. So they're saying increased human population decreases biodiversity. Now we need to explain that using all of this stuff going on here. All right, so let's have a look what's going on. So they've told you domestic animals such as cattle, sheep, and goats are farmed by humans to provide food. If you've got more humans, you're probably going to have more of that. And as a result, you're probably going to have less of the other wild ones like wildebeest and gazelle. And you could, like I said at the start, if you just had a quick look at that graph, well, yeah, I've just said that. So if we think about the evidence in support of it, well, your wild species are dropping. So from 1975 to 1985, they kind of were like you're good. Right, wildebeest and gazelle. I'm just lazy. I'm not. Um, but and as well as that, and you can see from about what like 1990 onwards, it's still decline while domestic animals increase. And that's true. You can see that. And I even pointed out at the start. So that's probably explaining that and supporting it. And you can give some numbers as well. Let's think about what the limitations are or what's not supporting it. If we're saying there's more population, there's going to be more cattle, then the other one should drop. So let's see if there's any problems with that. Well, if we look before here, there seems to be like, very little change in sort of the domestic animals, but they're still dropping here. So it might suggest that humans aren't actually the only reason. So before 1995, wild animals still decrease despite small changes in domestic animals. So that might suggest that humans aren't exactly just the only thing, but surely if it's humans, they're making more cattle. That's going to make them drop. So not that. What else have we got? So I mean, as well as that, these have all stabilized now. If it was truly that, then if we had less biodiversity, these would eventually be gone. So we still have gazelle and zebras, and they're relatively stable. And you give some numbers as well, which I've already had. So if we look, let's say, like since 1995, you can give some numbers as well from there. Other limitations, you need to think about the actual, I guess, procedure or experiment or things like that. So go through it line by line and think about what criticisms you can make. What can you talk rubbish on when this comes up? So Masamaras are blah blah blah, wildlife not very helpful. That's not something you can critique. That's also something you can't critique. What have they done? Several large mammals, such as gazelle, zebra, and wildebeest, and domestic animals. So one thing they're concluding that it will decrease biodiversity. We're only using in this experiment, not experiment, but procedure, or whatever you want to call it. We're only using three species of wild animal. Maybe there's way more. As well as that, it's not very reliable to look at just this on its own. So we're looking just at the raw numbers of them. We're not looking at the numbers of species and numbers of each species. So we haven't got a Simpsons index. The Simpsons index is probably one of your best measures of measuring biodiversity. As well as that, they're saying that if you have higher population, we're decreasing biodiversity. But we're concluding that because these are going up, these must be going down, which is a bit of a reach. So correlation doesn't always mean causation. And as well as that, we've even just said above as well that this is still dropping despite these. Sorry, not there. These are still kind of the same despite these also fluctuating a bit. So again, correlation does not equal causation. And as well as that, we have no statistical test to assess correlation between the domestic and wild animals. We need to do a statistical test to see is this truly like this is going up, that's kind of going down, there's maybe a possible link. Because it might just be by pure chance that's the case. Maintaining biodiversity in the Masai Mara is important. The following statements, the following are some statements. So people that visit Masai Mara, you see lions like lions, giraffes, and rhinoceros. Wild animals are sometimes killed by local people for food. Oh my god. Large herbivores such as wildebeest are the main source of food for large predators such as lions. Part of the area is used by farmers for grazing livestock. Soil erosion has occurred when trees have been removed by the humans to fuel clearing the land for grazing. In and around the Masai Mara, there are accommodation for over 7,000 tourists. The reasons for maintaining biodiversity can be grouped into three categories. So ecological, economic, and aesthetic. Fill the table to show which of these are used in an example, and you may use one letter more than once or not at all. So if you look at ecological, that's all to do with your biology and all of that kind of stuff, the interactions within organisms and all that kind of stuff. So large herbivores such as wildebeest, the main source, that's going to be your C. Economic is all to do with the money and the finances and stuff and all of that kind of stuff. So that's one of them. A is one of them, and D and F as well. So A, D, and F. And then aesthetic, well, again, it's A because you want it to look nice for these people to come and visit and stuff. So there you go. So in this circumstance, I won't draw it because just for the sake of time, but you need to draw essentially a suitable graph. In this case, the best thing you can do is a histogram. So to work out the frequency density of a histogram, which would go on this axis, you would do your frequency, so this 10 over the width of the class it's in, so over 100. And make sure you add the units as well. So this would be your frequency density per 100 millimeters. And then what you would do is on the bottom, you would just write them all out. So you would start with, what is that, 100 and 200, whatever the appropriate scale is, and so forth, and you draw bars that join each other like that, and that'll make your histogram like that. That's the best thing you can do with that kind of data. So now it says variation can be caused by genetic and environmental factors. Explain why your graph shows that at least some of the variation in the body length is caused by environmental factors. If you actually draw that, you get a bell-shaped curve. So if you have something that's controlled by a single gene and not environmental factors, it tends to be quite sharp, like one versus the other, or like nothing, right? If you have a nice continuous spread of results, like it's like that, that means that it's not just single genes, and it could be multiple genes interacting with each other alongside environmental factors. For example, if weight, like obesity, for example, was what's the word? Genetic thing, a solely genetic thing, then you'd have sharp obese, not obese. But we don't have that. People have varying weights. You don't just have one weight or another. That suggests that there's multiple genes involved, the environment is involved. And it's true. Well, like, diet and exercise and also history and genetics play a role. So it's all that. And that gives you that continuous data there rather than just set categories or like, straight up just like, one and nothing, right? So that's what it would be, is that you have a bell-shaped curve, or you could say normal distribution or bell-shaped, like graph. And that shows that the data is continuous, and that's what supports environmental factors being the case. So now it says it's important that commercial fishing is done in a sustainable way. So fish farms are one of the potential solutions to declining fish stocks. It lists 3 of the strategies that governments can use to increase the sustainability of commercial fishing. I mean, number one, you can just say that they could put quotas on to limit fishing. So that means you can't just go to town and absolutely rinse oceans like that. As well as that, what are the things you can do? So remember that when we're using fishing, when it comes to fishing, if you get like really tiny like mesh stuff, then all these other fish that you don't want can get caught as well, and that can screw up the food chain and all of that kind of stuff because other organisms don't get what they want. So you could use larger mesh nets. As well as that, maybe you could try and have specific areas where you can't fish. Also go for things that aren't endangered or rare. And also things like, for example, only fishing at specific times a year to allow them to actually reproduce and get back to where, to that sort of thing. So like, regular fishing seasons, for example. So those are a couple points you could say. International agreements are both an important approach to sustainability. Suggest why international agreements are particularly important in the case of sustainable fishing. Well, where are we getting fish from? In the oceans. And a lot of people we don't own an ocean. So that's pretty much it. The fish are in oceans between countries. So we internationally agree so that, like, I don't know, for example, I wanted to go and be like, all right, my country, boys, we're going to go get every single fish in the Atlantic Ocean. It's like, who's responsible for that? So it's just important to appreciate that if we internationally agree, then we can kind of be like, yo, don't do that and hold them accountable. So the abundance and distribution of plants can be surveyed in different ways. Some students wanted to survey abundance and distribution of plants in a small area of grass outside of school. The area was 20 by 20 in the size. So what they did is they laid 2 tape measures at right angles starting in the southeast corner of the grass area, and they used a random number generator to select x and y coordinates, and while facing north, they placed the left-hand corner of the quadrat to a point where the coordinates meet. Identify the species present in using a key, count the number of species in each present, record the table and information in a table, generate a new set of coordinates, and repeat 2 to 6 until quadrats have been sampled, 10 of them. The teacher said that this method would not allow them to measure the distribution of plant species, suggesting an improvement to the method that allowed them to do that. So don't read the question and be like, oh, I think it wants me to talk about this and start talking about, I don't know, random, sampling and this and that, blah, blah, blah. If you're looking at so abundance is simply just counting the numbers of things in an area. So like you can do here, 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 and whatever, and find like a mean and scale it up. Because you could probably find it per area centimeter squared, and then you times it out for the whole area. Distribution is where in this area are they mostly found. So the only way to really measure distribution is to start somewhere and then go across and see if you catch them. So for example, if we have like the ocean is here, and then we've got like grass, and then we've got woodland here. If we're looking at distribution, let's say all of these organisms tend to live here. If I randomly sample there, I'm only going to really get numbers, but I'm not going to know where. So the best thing to do is to start at one point, move across systematically, as in start like this. And the way we do that is by using a belt transect. So there you go. That's how you get distributions. You're figuring out where in that area. That's just not the number. So then it says identify a limitation with step 3 and explain why this limitation may affect the data. So it says while facing north, place the left-hand coordinate of a left-hand corner of a quadrat on the point where the coordinates meet. So they're ideally you want to put these quadrats down randomly in these coordinates and don't even think about it. If a person's looking at it and they're like, oh, I'm going to make these meet, and the left-hand corner of a quadrat where the coordinates meet. So for example, here and here, and then there and there, and here and here. So if they choose that, what if they decide, oh, I might just put it here because it looks good, or here, or there? So that's bias. So it could be that it could introduce bias, and we all do it, even subconsciously. And as well as that, it doesn't tell us what left side is it, the top left side or the bottom, like I just did there. So if we have these two coordinates meeting here, we don't know if it's going there, or like there, or even here as well. So it's really important to keep it consistent. Otherwise, maybe one person's subconsciously just doing that. So now it says, here's the results in the table. Calculate the Simpsons index of diversity for this student's data, and then use this formula. Whenever you see the sigma term, sigma, it's the sum of a total of something. Little n is the number of organisms of a species, and big N is the number of total number of organisms of all species. So what we need to do is we need to work out all the little bits and bobs for this. So we need to work out what n squared over n is for each of them, and then you would want, so n over n, for example, you could do, and then you would do n over n squared. So if we do that, so remember that little n is the number of a specific species, and then big N is all of them. So we need to work out what big N is first. So it's going to be 3 plus 7 plus 1 plus 26 plus 4 plus 3 plus 6. That gives us 50. So it's going to be 3 out of 50. And then you can square all of those as well. So if I just sort of awkwardly really do that, so 3 over 50 is what? And then if I do 1 over 50 squared, that's 4 times 10 to the 4. 10 to the minus 4, sorry. So then what I need to do is I need to work out what sigma n squared over n is. So I'm just taking that and squared it. So the sigma of all of that would be 0.3184. So then d would equal 1 minus your answer, which I hope to God is correct. So yeah, it is 0.6816. There you go. So then the students found grass species difficult to distinguish from one another, so they decided to record any grass species as grass. Explain how this decision might affect the calculated value. So think of it logically. Again, that is the number of a particular species over the number of all species. So if they group all the different types into just grass, what they're doing is that, remember that the total big N is going to stay the same. They just put it all as one big grass. So that means that little n is going to get higher, while big N, the total number of all species, is the same. So that means your sigma n over n all squared now starts to become bigger. Therefore, your d, since that's equals, it's 1 minus, well, that, your d becomes smaller. So that means the index of diversity is lower. So you can just explain it mathematically like that, and I'll do the job. On a Belgian field trip, the same students surveyed a large area of heather moorland. On this occasion, they did not use quadrats. Pairs of students were assigned a large group of moorland, and they rated the abundance of a species using the scale A is abundant, C is common, F is frequent, O is occasional, R is rare. One reason for the use of quadrats is inappropriate that many moorland plants are too large to fit in it. What other advantage is it? Well, I mean, it's just quicker. They can just quickly eyeball the entire area and be like, oh, there's a lot. Now, what's one limitation is that, well, it's subjective. I don't know what counts as like abundant or common. I don't know how to figure that one out. So, it might, it introduces sort of that variability. So now it says microorganisms can be used to produce a variety of food products. Microorganisms have simple nutrient requirements which help them reduce production costs. And there's 2 other advantages of using microorganisms in food production. So, for example, I don't know, if we're making like cheeses or something, for example, Parmesan cheese uses rennet from can't remember what animal it is. It's like cows. If you can do it with enzymes from bacteria instead, then you have no ethical issues there. So that's one of them. As well as that, if you're using microorganisms, you can genetically modify them to do what you want. So you can, do a bit of this and that and make an enzyme, for example. As well as that, there's what else could you do? It's non-seasonal as well, so you don't have to depend on them to sort of, be in season or whatever, or like in the right time of year to do something. You can also make them rapidly grow because bacteria, microorganisms, things like that rapidly replicate. And that means you can easily mean that you can just sort of upstage production pretty well. And as well as that, what else can you do? Those are the ones that I would say, to be honest. So that says yogurt is a food produced from milk using microorganisms. Yogurt production involves two bacteria, Lactobacillus whatever, and Streptococcus thermophilus. The bacteria converts lactose present into lactic acid. Lactic acid is an important contributor to the flavor of yogurt. Lactic acid also helps to give yogurt a longer shelf life than milk. So just how lactic acid helps to extend the shelf life of yogurt. So again, good thing to do is to just have a second to understand what you need to explain. What's the end consequence? Longer shelf life. And then how? Lactic acid, right? Then you need to think, well, what's going to screw up the shelf life of yogurt? I mean, if you think about it, what's going to make them go? Well, bacteria are going to make it all go off. So that means lactic acid. I mean, how does lactic acid affect bacteria? Oh, well, it lowers pH. Therefore, it could denature bacterial enzymes, or it could just make them less effective because it might not be their optimum. So both bacteria also break down some of its protein, casein, which is present in milk. Name the product of protein breakdown and describe the type of reaction that takes place. So what you're making is, remember, you're breaking the peptide bonds between them, so you're going to make amino acids eventually, and that's done with hydrolysis, the opposite of The microorganism that's used to make mycoprotein is a fungus, and here's the diagram of a fermenter. So what have we got going on here? We've got ammonia and air coming in here, glucose coming in here, the products collected there, we've got a cold water cooling system, and then we've got carbon dioxide out there. So it's really important just to try and think, why is that all there? What's the point? Don't just look at the diagram, jump to the question, try and answer it because you think that's what, and then be like, oh, I think they're asking me this. Just make sense of what's going on here. Okay, so the types of fermentation you've got are batch fermentation. So you put microorganisms in a fixed volume of medium, the stuff gets used up, biomass and waste products are made, and then once it's done, you sort of you stop the process before you start to get it too much, and then you just clean the system, sterilize it, and then start a new batch. Or continuous is where you completely just keep things cycling. So you keep adding the nutrients and stuff like that, and then you continually remove the material within the system and separate all the stuff, but you keep the volume within the system the same. So you're taking stuff out in one, putting it back in. Here you're keeping everything in. It's just cycling. So in this case, what's going on here is that they are putting in ammonia, air in, glucose in, carbon dioxide in, and it's a continuous just cycle, and all the stuff that's coming out here is product. So by definition, it is continuous. And you could say all of that, but there's just an outlet for product collect. Because remember, in batches, they don't have an outlet. They just take the entire thing out, do all of the bits that they need to grab it, and then put it back in, and start again. So just why a cooling system is necessary. So you have something where you've got fungus making mycoprotein in the system, and they've given you a cold water system. So you need to think, how's that going to screw up or mess with or make things better for the fungus to sort of make the mycoprotein? So ideally, you'd be like, higher temperature is better. What the hell? Why would they use a cooler? And then you need to think a bit more beyond that. Well, if you left these fungus to do their job, everything in the universe, when it's doing respiration, releases energy, and that's heat energy. So respiration releases heat. If the system gets too hot, this could, denature enzymes and proteins. As well as that, it could also mean that you could get pathogenic bacteria and contaminated other organisms as well, which can compete and cause problems too. So it's just taking that extra. So the air inlet provides the fungus with oxygen for respiration and ammonia. So just to explain why the fungus is provided with ammonia. So it's given fungus plus ammonia, and now it says why. Now you need to think, well, in your head you should be thinking, oh, in that case I need to figure out what the hell they actually use ammonia for. So the only thing that funguses and living things actually use it for is that it must be a source of nitrogen, and it's used to produce amines. There's absolutely no other sort of obvious thing that says why they would have ammonia. There you go. Next. Mycoprotein foods contain protein. The presence of protein in a solution can be determined by a biuret reagent. In the presence of the protein, the biuret reagent turns from blue to violet. I'm stuttering a lot today, sorry. A colorimeter can be used along with a biuret reagent to determine the concentration of protein in a solution. Outline how a colorimeter can be used to determine the concentration of a protein in a solution. They've told you that, again, colorimeter on a solution can find the protein concentration. So there's a couple gaps here, because you should appreciate that a colorimeter can simply measure how much light is absorbed. So you need to think, how can I measure light absorption and then work out protein concentrations? So the only thing that should come to mind is that you can produce a calibration curve. So here you would have the absorption of light, and you would have the concentrations, right? And it could probably produce this, or probably go down this, and then you could, I guess, put a sample in, and you do that, and you find out, sorry, the other way around. You put a sample in, and then you can find its concentration like that. So then, that's called the calibration curve. So it could be a straight line or curve or whatever it is. So then you need to outline how you would do this. So what you would do is you would, first of all, you would put samples of known concentrations of protein with the bioreactor reagent. I'm just going to write bio reagent into the colorimeter. You should zero it before use. And then you should record the absorbances of each mixture, or sorry, each concentration of syrups, and plot a graph of concentration against absorbance. And then what you can do is you measure the test sample that you now have, and then you just read off the graph. So read off concentration from graph with from graph with absorbent. That's it. That's how you do them. And actually, to be honest, colorimeters, you should probably add like a filter as well on it to make sure that only light of that color is coming through. So if they've told you it's going from what, like a blue to violet, then you should probably filter it so only blue, sorry, so that blue's out and violet's only been measured. So you should probably add that too. That's how you do it. So again, it's looking at colorimeter and somehow you're able to find protein concentrations. The only thing that's going to be used and linked to it is the absorbance. State one alternative method for determining the concentration of protein in a solution. So biosensors. Because how biosensors work is that they can use bits of like biological components and things like that to determine, your sort of concentration or like things like glucose. So, for example, it could be that there's like an enzyme or an antibody or some single-stranded DNA stuck to a surface, and then it will, like glucose will bind to it as it passes along. Or you could do something like a transducer. So, for example, if you have changes in pH, what it can do is it can make a dye or some sort of thing be released from a test strip, and then it generates an electric current, and then it can, provide data like that. So that's quite good of being able to measure things as well. Because what they'll do is they'll make their own calibration, and then, for example, if there's like a certain current, then they will have calibrated it with known solutions themselves, and then the sensor would be like, oh, well, if it's got a pH of this, then it's probably that concentrate. So now it says complete this following sentences about the controller body plan using the appropriate terms. So a body plan is where you have a cross section of an organism. So for example, a slice through it, and it will show you how like the organism's tissues and stuff are layered out. Now, the reason that happens, well, so the reason we have body plans is because, we've got, again, organisms of all kinds of shapes and sizes and all kinds of things like that. We still have, like, again, we've all kind of sort of emerged from one single little organism and so forth. We all have quite similar genes and, similar things in us, but we don't all look the same. The reason for that must be that we've got, like, expressions of different genes and controls of different genes and so forth. Need to know about homeobox genes and also then again Hox genes as a result. So in homeobox genes, it's a section of DNA where you have where you code for a part of a protein that's highly conserved. Where again, what that means is it's very similar between different organisms. What that does then is there's a part of a protein called a homeodomain, which binds to DNA and it can either turn genes on or off. And when you turn a gene on or off, it means you are either expressing it, so you're transcribing and translating it, or you're not. An example would be PAX6. One of the homeobox genes is PAX6, and what happens is if you have a mutation that stops it from working, you can have underdevelopment of the retina, which can cause blindness. And as well as that, it happens in other organisms. So, for example, I think like mice can also have blindness as well if a mutation happens. So that means that PAX6 is probably a homeo gene that helps development of eyes across different species. Not necessarily, but it suggests it. Hox genes are a type of homeobox gene that's only in animals, and therefore to do with sort of putting things around, as in like positioning different parts of the body. We have gene clusters, which is where these Hox genes are found, and we have 4 different clusters. We have 39 Hox genes, and we think that it's all to do with one sort of homeobox gene that came along and then it's just sort of duplicated over time and had loads of different mutations, and that's what's given us our little spreading out things like that. So anyway, to this question, it now says, body plan is under genetic, and we need to think what will have caused those different things being expressed and not expressed, because we don't always express all of them, as with all genes in the universe, and we all look a bit different. So it's got to be environmental as well. Whenever you have a gene that is just on or off, oh my god, why can't I write? Sorry. If you have a gene and it's on, you should have it. If it's off, you should not have it. If it's not doing that, then it's either an environmental thing or you've got something like epigenetic marks or something like that controlling it as well. So generally speaking, it's intergenetic and environmental control as well. And then internal and external something can influence the expression of these genes that regulate the cell cycle. So anything that's changing something that's going on inside you, like a behavior of some sort, is a stimulus. So stimuli, a change in an internal and external environment. Such genes can promote and inhibit programmed cell death, known as apoptosis. During programmed cell death, something digests the cell contents and the products are removed by something. Enzymes. And we need to get rid of all this gunk, phagocytosis. Then it says, state the name and type of gene responsible for controlling body plan in multicellular organisms. I just said homeobox. So sorry. Well, homeobox is allowed, but Hox specifically. And there you go. That's that end of the paper. Whoops. Thank you for watching. Please like, share, and subscribe. You know the drill. And please tell others about this channel and help your friends out, and you know the drill.